Hello everyone, I'm Shurosri Pal. We are solving this question from GATE examination 2013, paper Biochemistry, question number 10. The question states, in different types of tissue transplantations, the rate of graft rejection in decreasing order is A. Isograft greater than xenograft greater than allograft. B. Allograft greater than isograft greater than xenograft. C. Xenograft greater than autograft greater than allograft. And D. Xenograft greater than allograft greater than isograft. For more information regarding tissue transplantation and graft versus host reaction, please follow this NPTEL course Immunology. This is the lecture Transplantation or Graft versus Host Reaction. The link is also provided here. We have to talk about different types of transplantation. So the first one is autograft. Autograft is when transplantation is carried out from one part of the body to another. So inside the same individual. So this is for the same individual. This is called autograft. What is isograft? Isograft is between genetically identical individuals. So these genetically identical individuals should be twins and they should be monozygotic twins um, who were born from the same zygote. Then coming to allograft. Allograft is when transplantation is carried out between different members of the same species. So it basically means between two different individuals. Between two different individuals. Like Mr. Smith to Mr. Jones. So we are two different people, and between them, if transplantation is carried out, that is called allograft. The next one is xenograft between members of different species. Like for example, pig to human. So it is between different species. Now, this transplantation is actually a difficult process. Tissue transplantation has a major problem with it, which is the graft rejection. So graft rejection is when the body rejects that particular graft. It can be a tissue, it can be an organ, it can be several things also. So when this graft is rejected from the body, that means this grafted organ or this transplanted organ cannot survive in the body. So why does this, first of all, why does this graft rejection occur? So, graft rejection is an immunological response which is mediated primarily by the T cells. The basic rules of tissue grafting were first elucidated by skin transplantation between inbred strains of mice. So, skin can be grafted with 100% success between different sites on the same animal or person, which is autograft. This is something that we discussed first. Autograft is when skin from one individual is replaced. So from one part of the body, it is replaced onto some other part. So that is usually 100% success or between genetically identical animals or people, which is a syngenic graft or an isograft, right? But the problem is when skin is grafted between unrelated or allogenic individual, that is called an allograft. The graft initially survives, but is then rejected about 10 to 13 days after the grafting. So, in case of an allograft, the graft is rejected after 10 to 13 days. Now, why does this happen? The basic rules of tissue grafting were first elucidated. Uh, Sorry, this is just a repetition of the same thing. Actually, what happens is that antigens that differ between members of the same species are known as alloantigens. And an immune response against such antigens is known as an alloreactive response. So when antigens uh, that differ between members of the same species, so that means antigens that differ between different individuals, they are called the alloantigens. And when there is an immune response against such antigen, this is called an alloreactive response. Now, when donor and recipient differ at MHC, so remember that major histocompatibility complex, these are the these are also called MHC, and these molecules actually can differ between different individuals because we know that MHC is polygenic and polymorphic. So there is a lot of variation of MHC in different individuals. So often it happens that the donor and recipient they differ at the MHC. So when this MHC is different between the donor and the recipient, then an alloreactive immune response is directed at the non-self allogenic MHC molecule or molecules on the graft. So, therefore, this graft rejection is primarily due to the role of the major histocompatibility complex or MHC. Because the donor and the recipient they differ at the MHC molecules, so in the recipient's body, the immune response or the immune system of the recipient detects these foreign MHCs on the graft as alloantigens and it generates an alloreactive immune response against those MHC molecules. So, in most tissues, they are predominantly MHC class 1 molecules. Once the recipient has rejected a graft or a particular MHC type, any further graft bearing the same non-self MHC molecule will be rapidly rejected in a second response. So, the frequency of T cells specific for any non-self MHC molecule is relatively high, making differences at MHC loci the most potent trigger of initial graft rejections. So, always remember this that graft rejection is due to the non-self MHC molecules that are present on the graft, which is not same as that of the MHCs of the recipient, and therefore the recipient's body's immune system it does not allow the non-self MHC molecule and it generates an immune response that destroys these non-self MHC molecules and so destroys the graft. So here it is an example of an acute rejection of a kidney graft through the direct pathway of allorecognition. So here this is a kidney graft with the dendritic cells. Now the dendritic cells migrate to the lymph node and the spleen via blood where they activate the effector T cells. Now, these dendritic cells from the graft, they migrate to the lymph node and the spleen via blood, and they activate the response of the effector T cells. Now, effector T cells migrate to the graft via the blood these effector T cells now come to the graft, and then graft is destroyed by effector T cells because these T cells are the cells that recognize the MHC molecules as alloantigens or non-self. So therefore, the effector T cells destroy these non-self MHC molecules, and the graft is actually destroyed. So this is the pathway of allorecognition or graft rejection. Now, when donor and recipient are identical at MHC also, there can also be graft rejection. So what is actually done is that before transplantation, the donor and recipient, they're matched for their MHC molecules, right? But it was observed that even if the donor and recipient, they match at their MHC molecules, then also these two individuals might differ at other genetic loci, and therefore graft rejection can occur. It will be not as rapid, but if it is left unchecked, it will destroy the graft. Now, that is the reason that grafts between HLA identical siblings or MHC identical siblings would be rejected without immunosuppressive treatment. So, usually, what is done for this kind of grafts or tissue transplantations, that the recipient is put under immunosuppressive drugs so that the immune system of the individual can be suppressed or the so that it can delay the response of the effector T cells and therefore the effector T cells cannot come and destroy the graft. Right, so the MHC class 1 and class 2 molecules bind and present a large selection of peptides which are derived from cell proteins made in the cell. And if these proteins are polymorphic, then different peptides will be produced from them in different members of a species. Such proteins can also be recognized as minor histocompatibility antigens, right? So instead of major histocompatibility complex, there are also minor histocompatibility antigens, which also differ between different individuals because they are polymorphic. So therefore, even if the donor and the recipient they match at their MHC, there are other antigens like the minor histocompatibility antigens that will also generate immune response. So the graft rejection will occur ultimately. It will not be very rapid, but it will not be rapid. But the problem is that it will not be rapid, but uh, then again, the graft rejection will ultimately occur. So we need to put the patient on immunosuppressive treatment. Now, now coming to the question of graft rejection and what are the chances of graft rejection. So therefore, we can understand that when there is autograft, there is 100% success because it's between the same individual and there is no chance of mismatch of MHCs or any other antigens. So in case of autograft, the chance of graft rejection is minimal or it's the lowest, right? So the rate of graft rejection in decreasing order, so which means that um, at the last or the most um, or the least chance of graft rejection will be in case of autograft, right? Now, coming to the next thing that is the isograft. Isograft will obviously have a higher chance of graft rejection than autograft, okay? But again, it will be lesser than allograft and that will be again lesser than xenograft. So obviously xenograft will have a highest chance of graft rejection because these two individuals are belonging to the same to the different species. Actually, they do not belong to the same species. So therefore, xenograft will have a higher chance of graft rejection. So which means that the least chance of graft rejection will be for autograft. Okay, so the least chance is for autograft. Now, which will be followed by isograft because isograft is between two genetically identical individuals and if like there will also be there can also be graft rejection between genetically identical individuals in case they do not have the same minor histocompatibility antigens, but um, the chances are pretty less. So higher than autograft will be isograft. Now, higher than isograft, the rate of graft rejection will be observed in allograft. Allograft is between two different individuals, and there will be high chances that these two different individuals they do not have the same MHC molecules. So, in case of an allograft, the chance of graft rejection is higher than isograft. So here we will write greater than isograft, the chance of graft rejection is higher in allograft. And of course, the highest chance of graft rejection will be for xenograft because it is between two different um, two organisms which belong to two different species, right? So therefore, the highest chance of graft rejection will be in case of xenograft. Okay, so according to this, our correct option will be D, xenograft greater than allograft greater than isograft, right? and autograft is not there in this option. So our correct answer is option D. And that's it.